Good afternoon, everyone, and welcome to Live from the Lab at La Jolla Institute for Immunology. I'm Dr. Sonia Sharma, Associate Professor here at LJI and Director of our Center for Sex-Based Differences in the Immune System. It's a real pleasure to have you with us today. Since its launch in 2020, Live from the Lab has brought nearly 20 virtual sessions to life, each one designed to open the doors of our research labs in Southern California to audiences across the country and around the globe. We're deeply proud of this series because it turns what can feel like a very specialized scientific world into an accessible, welcoming space for anyone who's curious about how the immune system shapes human health. I had the honor of being the featured presenter at this year's Live from the Lab, which was sponsored by the Global Autoimmune Institute, where I shared my team's work on the earliest immune signals that drive autoimmune disease. These webinars are most often moderated by our president and CEO, Dr. Erica Ullman-Sapphire. But today, I'm so pleased to step into the moderator's seat in my role as director of the Center of Sex-Based Differences in the Immune System. It's a real privilege to highlight some of the promising philanthropy-fueled research that's happening in this space right now here at LJI. At LJI, we believe that understanding the immune system, this beautifully complex, ever-adapting network, is the key to solving some of the most urgent health challenges that face us today. From cancer to neurodegeneration to chronic inflammatory diseases and infectious diseases, immunity sits at the center of human health. Yet for too long, one of the most powerful variables that shapes immunity has really been overlooked, and that is biological sex. Every cell in our bodies carries sex chromosomes, XX in women, XX in men, XY in men. And those chromosomes hold many genes that influence immunity, that influence your immune system. The X chromosome alone contains dozens of immune-related genes, and women carry 2 copies. Beyond genetics, the sex hormones, estrogen, progesterone, testosterone, reach nearly every immune cell, altering how those cells respond to infection, injury, and disease. New studies are showing that women launch more than 1,000 genes differently than men, even outside of X and Y chromosomes. These are not subtle differences. They profoundly influence how diseases develop, progress, and even how they respond to treatments. And yet, for decades, much of modern medicine has relied on a one-size-fits-all approach. This is built from research that often excluded women or failed to account meaningfully for biological sex. The result? Therapies that might not work in the same way comparing women and men, missed opportunities for prevention, and blind spots in how we treat disease today in the clinic. Our Center for Sex-Based Differences in the Immune System was created to close this gap at the most fundamental levels. Here, scientists are now mapping the immune system cell by cell, integrating genomic, epigenetic, metabolic data to understand how sex shapes immune function and immune disease. We're asking how hormone levels, pregnancy, life stage, genetics alters the body's immune defenses, and how this knowledge can lead to more precise, effective, and equitable treatments for everybody. Today, it's such a privilege to shine a light on 2 young scientists who embody the mission of the center. Both of these young scientists are recipients of funding from the Tullie and Rickey family's Spark Award for Innovations in Immunology, a program at LJI designed to give early career scientists their very first foothold for innovative ideas that haven't yet attracted federal funding. Spark offers modest 8-month seed awards to support these investigators as they generate the critical early data that's needed to compete for a larger sustained support. Now in its 9th year, the SPARK program exists thanks to the sustained generosity of LJI board directors Tom Tullie and Dave Rickey and their families, whose commitment has ensured the program's growth and longevity. Since its start, more than 240 donor families have joined the Tullies and the Rickeys in supporting over 50 innovative projects. Each year, early career investigators from across LJI submit their proposals. The finalists then pitch their ideas to LJI leadership and the donors. Those selected receive funding to begin their proof of concept studies. This is such an important early step towards their independent research career and fuels the most innovative ideas. Today, Dr. Amparo Martínez Pérez will begin by sharing her SPARK-funded project, which introduces a novel approach to studying immune cells found in menstrual blood, a powerful non-invasive window into diseases like endometriosis, which have long lacked effective diagnostics and treatments. Her work could open new paths for earlier disease detection and better care. Next, Laura Hinojosa will share her SPARK-funded project, which maps how hormonal contraceptives influence immune cells, bringing new insights into a class of medications long overlooked in immune research, but we think are going to have a big impact. Her work could open new paths for earlier disease detection and better care, while building the knowledge needed for more personalized, evidence-based care in the future. Together, both of these projects are challenging historic blind spots in research, pushing us towards a more equitable and precise understanding of the immune system for women and for men. As you listen, I encourage you to think about the individuality of immunity, the idea that our genetic background, our hormone levels, and yes, our biological sex all shape how immune defenses rise or how they falter. This understanding is the foundation for the next generation of personalized medicine. Now, before we dive into the presentations, I just want to take a brief moment to introduce LJI for those of you who might be joining us for the first time. LJI is an independent, nonprofit research institute that is dedicated solely to understanding the intricacies of the immune system. Next slide, please. Your immune system drives nearly every major disease, from cancer and infections to chronic inflammatory disorders, and the differences in how men and women experience illness. To tackle this breadth, LJI is organized into 4 different research centers, each one designed to focus expertise and foster collaborations between our scientists. Next slide, please. Our more than 200 scientists are working right now to harness the power of the immune system to develop better vaccines, new immunotherapies for a range of diseases, and groundbreaking treatments for conditions that currently might not have any treatment or cure. LJI is purpose-built to surround our scientists with expert administrative support, allowing them to dedicate all of their energy and talent to discovery. This deliberate structure is one of the key ways LJI stands apart from its peers. Next slide, please. It's important for me to stress that the discoveries made here, they don't just stay in the lab. They move into clinical applications. They help reshape how diseases are treated, and in many cases prevented. We're honored to be a quiet force behind the innovations and tools that impact you and your loved ones, how they are diagnosed, treated, and cared for during life's hardest health moments. We're actively advancing new diagnostics and therapies every single day, creating the innovations patients depend on today and will depend on in the future. Next slide, please. And with that, it's my absolute pleasure to introduce one of today's featured speakers, Dr. Amparo Martínez Pérez, who is a postdoctoral fellow in the Weisskopf Lab here at LJI. Amparo's scientific journey began in Spain, where she earned her Ph.D. in immunology, studying how vaccines and infections shape the lungs' immune defenses. She joined LJI's Weisskopf Lab in 2023, which is known worldwide for its work on T-cell responses to emerging viruses. The lab has made important discoveries focused on how men and women's immune systems respond differently to these types of infections. And these are insights that are currently shaping vaccine design and immunotherapy strategies. Building on her background, as well as her lab's specialized expertise, Amparo designed a philanthropically funded project that's looking at specialized immune cells to provide new insights for women's health conditions like endometriosis, a disease that affects millions, yet still lacks reliable diagnostics or effective treatments. To give you a sense of program flow following Amparo's presentation, we'll later be joined live from the lab by Laura Hinojosa, and then we'll open the floor to questions, which I'll moderate. Please feel free to submit your questions at any time through the Zoom Q&A feature, and we'll address as many as we can. Amparo, we're so grateful to have you with us today and learn more about your innovative project. I'll turn the program over to you now.
Thank you, Sonia. Okay, thank you. Hi, everyone. It is a real pleasure to be here today and to share with you some of our latest work. But before starting, I want to acknowledge all the people that are involved and have been involved in this project, because normally science is always teamwork. Starting with all the members of the LGI clinical core that have been in charge of recruiting the donors, collecting the samples. Hayley, Simon, my lab mates from the Weisskopf lab, and special thanks to my PI, Daniela Weisskopf. And as Sonia mentioned, this project has been possible thanks to the existence of the Spark Awards program, and special thanks to the Rosemary Crematory Foundation, who funded this specific work. And last but not least, of course, a big thanks to all the volunteers who donated for this. I'm going to start my talk by very briefly explaining the components of the adaptive immune system, which is the part of the immune system that has immunological memory. Now, we all have probably heard about antibodies, which are a type of molecule that are produced by a cell called B cells. But there is another component of the adaptive immune response equally important, and these are T cells. And they just act in a different way than B cells. Instead of producing antibodies, they defend the body by secreting some molecules that coordinate the rest of the immune response, or even by directly attacking and killing either infected or tumorigenic cells. Now, I'm going to continue talking about T cells, and I'm going to highlight three main features of them: memory, specificity, and location. Memory. We have already said that these cells have immunological memory, which is classically defined as their ability to respond faster and more efficiently to a pathogen that they have previously encountered. Second, specificity, meaning that each one of these T cells will only be able to recognize one specific type of pathogen. And third, location, because these cells can be found either circulating the body through peripheral blood, or they can be found residing in the tissues where they first encountered the pathogen, becoming a faster and more efficient way of responding to a second infection. Now, if we put everything that I've previously said together, it makes sense that for different tissues of the body, we have a different distribution of these pathogen-specific or antigen-specific T cell responses, depending on where infections have been taking place through the body. And we scientists have studied the immune system, the immune cells in blood for the longest time. Also because it's fairly easy to access it and to have peripheral blood samples. However, over the last decade, it has become more and more obvious the important role of these resident memory cells and how crucial it is to understand how they act in order to comprehend if we are going to be protected or not towards new infections. But you can imagine that studying this in the human body is complicated, requiring very invasive procedures with complex techniques and even expensive. But there's been some work that has been done in different organs of the body. And Sonia was mentioning about sex bias difference and the big gaps in knowledge that we have. And of course, one of the less studied tissues in the body is the female reproductive tract. Now, from an immunological point of view, the female reproductive tract is a really interesting site. It's a barrier site because it's a port of entry to the body and it commonly has to face pathogens. But it must balance a defense against these bad pathogens versus tolerance to the fetus in case of pregnancy. We know that the fetus has half of its genetic material is different from the mother, and we don't want to attack it and destroying things thinking that is a threat. Therefore, the whole system must balance a very tight and complex immunological regulation that we still cannot comprehend. Now, we think that T cells play a crucial role in all the defense of the tissue, but there is actually very little that we really know about their role in infections, cancer immunity, autoimmunity, or even fertility issues. But we have the same problem as before, right? How do we study without relying on very invasive procedures? Well, in this case, we can use another unique feature of this tissue. We know that in reproductive age women, during the human menstrual cycle, the endometrium or the lining of the uterus thickens every month in order to provide support for a potential fetus. Now, when pregnancy doesn't occur, this endometrium gets shed and discarded in the form of menstrual blood or menstrual effluent. Now, this menstrual blood is not only composed of structural cells. There are also a lot of immune cells that were previously located in the endometrium. And of them, up to 40% are T cells. And when I started researching about this, I got shocked of how little did we actually know about this T cell that got every month discarded in menstrual effluent. And that's when this project came into life. We wanted to use menstrual effluent as a non-invasive and inexpensive way to comprehend T cell activity in the uterus, to be able to access this T cell and interrogate and investigate what is their ultimate role in the endometrium. But the first question that we had to answer in this project is, can we actually obtain viable T cells from menstrual effluent? Because in order to perform our T cell analysis, we need the T cells to be alive for at least a couple of days. And thanks to the financial support of the SPARK project, we started this work. We started recruiting patients, working through the logistics of establishing this absolutely new study here at LDI. And the moment that the first samples start arriving, we've been working a lot on protocol optimization because we realize how this type of sample is completely different from peripheral blood, which is what we are mostly used to. How it can also vary, but be really heterogeneous from one donor to another, or even from one menstrual cycle to another. And of course, everything making sure that then the cells had enough quality for our subsequent analysis of interest. But the good news is that at the end, we end up finding these viable cells, and I'm really excited to show you how they look like in flow cytometry. Well, flow cytometry is a methodology that we normally use for characterizing the cells in a sample, okay? Very simplistic explain. Each one of these dots represent one cell, and the Y and X axis represent two different features. not important right now. What I want you to focus is on this highlighted circular area over here. This is how a peripheral blood sample run through the flow cytometry normally looks like. And we know that in this area is where lymphocytes or T cells should fall. So there's a big chance that anything that is in this area is a T cell. When we run our menstrual effluent, now we find also this really nice, a different proportion, but still a really nice and existing population of T cells here. And then, of course, we did subsequent analysis to make sure that these cells were alive and that they were actually T cells with further markers. But I'm not going to show this today. In any case, this was really exciting when we were, for the first time, looking at this in the cytometer. And then we also started investigating a little bit more about these cells, and we realized that they are not quite the same, the T cells that we found in menstrual effluent versus in peripheral blood. For example, measuring the ratios of the subtypes CD8 or CD4, we are seeing that in menstrual effluent there is, on average, a higher proportion of CD8 compared to peripheral blood. And also, by measuring the expression of tissue residency markers, there are some proteins that can be expressed on the cell to help them stick to the tissue, get attached to the tissue instead of getting back to circulating through blood. We were also seeing for the first time these very nice and clear populations expressing these tissue markers, which was also very encouraging. And we have never ever seen this in peripheral blood. So we can see how the two samples are different in some aspects. But the real question that we wanted to answer was, okay, can we kind of determine the role of these T cells? Can we identify what is their antigen or pathogen specificity, right? So for doing that, what we need to do is culture these cells with a peptidic mix of different our pathogens of interest, stimulate them for between 6 and 24 hours, and then trying to see if we can find any activation markers, production of cytokine that let us know that these cells have identified and respond to this specific stimuli. Now, this is going to be all menstrual effluent samples, and this is how a negative response look like. Now, if we are, in case of finding positive response, we will expect these cells to fall into this upper left quadrant, depending on the stimuli that we are adding here. And when we started analyzing them, we started seeing, in certain cases, the existence of these antigen-specific cells that, of course, are also very donor-dependent, right? Based on our history, in our medical infection history. For example, in this donor, let's say A, we see that they don't have any immunological memory to cytomegalovirus, but a nice response to Epstein-Barr virus. Meanwhile, in donor B, you can see even a higher cytomegalovirus response, probably reflect that they have had a persistent chronic infection. Then there is a lot of T cells that have been created to combat this virus. So we were really also excited to find this data that our approach was finally working, and we were able to determine the specificity of these T cells. And by doing this with a lot of donors, analyzing longitudinal samples, also comparing them to peripheral blood, we are starting the first atlas of antigen-specific T cells in menstrual effluent. And with this atlas, we want to answer, okay, what are these endometrial T cells recognizing in terms of specificity and also quantity? How many of these cells do we have? How do they respond to those antigens? Because I am showing here only some activation markers, but we are measuring a lot of other markers because T cells can act in different ways, secreting different type of molecules, depend on the context. And lastly, and more importantly, are they imbalanced or dysfunctional in disease? Why these cells can sometimes not work properly and we still get sick. And of course, having potential diagnosis or therapy implications. And we are just starting applying this tool to the study of endometriosis, which is a really common, fascinating, and still very unknown disease. But we hope and we believe that our tool can be used to investigate the endometrial context in a lot of different diseases, like, of course, infections, cancer, autoimmunity, or fertility. Investigate how the T cells are acting in a bad or a good way in any of these female reproductive tract conditions. And we are currently looking for financial opportunities for any of these areas. So this is my last slide. You almost made it to the end of my talk. Bear with me. I want to finish by giving some take-home messages that are that we are able to study endometrial T cells through menstrual effluent, and we are doing that by measuring the numbers, dynamic functionality of these antigen-specific T cells. But the final goal is to identify differences or dysfunction in these T cells linked to women's health and other reproductive disease. But in order to keep this type of project, mine or the other type of project going, we need to advocate for change in the current funding. Women's health and sex-based differences are field just starting to be explored, and it's so full of interesting questions that shockingly we still have not been able to answer. So we as a society has to be aware of these big gaps in knowledge, speak about them, talk to our friends and family about it, and ultimately demand for it to change and to put more resources into investigating all of this. And with that, thank you so much for listening. It's been a pleasure, and I'll see you at the Q&A section.
Thank you so much, Amparo, for sharing your fascinating work and for helping us better understand the reproductive immune system and its role in diseases like endometriosis. It's an area that we've actually had very little data in the past, and your project is filling a critical gap that can inform more personalized approaches to women's health. Next, we're eager to hear from Laura Hinojosa. Laura is a graduate student at UC San Diego who's working in the iLab, where scientists used advanced computational tools to study how genes are turned off and turned on, and how those gene switches influence the immune system in both health and disease. Their work is uncovering the genetic and molecular patterns that shape how the body responds to infection, inflammation, and other immune-related conditions. Many Spark Award recipients hold MD or Ph.D. degrees. Sometimes they hold both. So it's especially noteworthy that Laura is one of the few graduate students who's been selected for this highly competitive award. And now, without any further ado, it's my pleasure to turn the program over to Laura.
Thank you so much, Sonia. And thank you for inviting me to share a little bit about my most recent work here. Right. So, as Sonia mentioned, I'm a graduate student at UCSD, and currently I'm trying to answer this question, which is how does hormonal birth control shape the immune system? And this is work that was made possible by the generosity of the Tullie and Rickey family's Spark Awards for Innovation in Immunology. So, first of all, we have talked a lot today about how men and women are different and how our immune systems are different. And we could say that there's these three main players that make that happen is our differences in genetics. We have different sex chromosomes. There's a bunch of environmental factors when we're growing up and developing that will also influence how our immune system behaves. And ultimately, sex hormones play really a key role in regulating our immune system. And in fact, most of the cells in our bodies have the ability to detect these sex hormones that are circulating in our bloodstream, and they can react accordingly. And this is especially important in our immune system. If we were to zoom in into a T cell, for example, we can see that when estrogen comes into contact with estrogen receptors, either on the cell membrane or inside of the cell, this is going to trigger a series of molecular cascades that will ultimately result in the DNA being accessed and certain genes being activated or repressed. And which genes are being launched by our cells determines how our cell is functioning. So in other words, sex hormone can control immune function. And we've also talked a lot about today about how important it is to have a carefully regulated immune system. If you have a suppressed immune system, you are more susceptible to cancer or infection. And if it's overactive, you could develop autoimmune diseases or inflammation disorders. So you really want to have an immune system that will be able to recognize the pathogens and mount an appropriate response to them. And sex hormones playing such a big role in these becomes very evident for probably everyone listening, either by first-hand or second-hand experience, when we looked at the menstrual cycle. So in reproductive aged women, on average every 28 days, ovulation occurs. And this is a process that is tightly regulated by really surprising fluctuations in these sex hormones. Here I'm showing two of the main players, estrogen and progesterone, which usually get most of the attentions, but they're not the only things that are regulating this process. There's many other hormones that they interact with as well. And what happens when these hormones fluctuate cyclically is that alongside with regulating this process of ovulation, we also have other things that are changing in our bodies. And I don't think I have to go into the details. We're all probably aware. But what's really interesting in terms of the immune system is that immune function is also being affected. And one of the ways that we can see that is in a lot of patients with autoimmune diseases, such as lupus, for example, sometimes symptoms worsen right before menstruation, for example. And what we're particularly interested in looking at is birth control, because hormonal birth control works by having synthetic sex hormones that disrupt these cyclic fluctuations. And they come in two flavors. They can either be combined, which is when you have both an estrogen and a progestin, or they can be progestin only. And in the combined case, they essentially work by tricking the body into thinking ovulation has already occurred and thus preventing pregnancy. In progestin only cases, it also thickens the cervical mucus and has other mechanisms by which it works. But in general, if we are having differences in these sex hormone fluctuations, how does taking birth control change our immune system? How does it shape our immune response or our immune cell populations? And we know some things from the literature. So, first of all, it's a really effective contraceptive. And historically, it has also been a really powerful tool for managing a lot of gynecological conditions, such as polycystic ovary syndrome. And it has a wide variety of side effects that vary from person to person. And in terms of autoimmune disease risk, we have seen in some longitudinal studies that taking birth control can actually affect your risk of developing certain autoimmune diseases. It can decrease the risk of rheumatoid arthritis, for example, or might increase the risk of certain autoimmune disorders of the skin. And then there's other cases, such as lupus, which I added here, that are actually quite controversial still. And all of these findings are extremely dependent to the patient, the type of hormonal birth control that they're taking, how long they take it for, how old they are, a lot of confounding factors. And there's a lot of studies that contradict each other. And one of the reasons why it has been kind of hard to have a clear guideline as to what to do when you have autoimmune diseases or how exactly this is affecting our immune cells is because despite having had access to hormonal birth control for over 60 years, there's actually a really small amount of studies that have been done in trying to understand at the molecular level what is going on. It is often not a variable in studies, and there's multiple confounding factors, such as the specific brand or formulation of the birth control that you're taking, which can affect the effects. And so what we aimed to do here is to have the first study that uses single-cell multiomics to evaluate the systemic effects of taking oral hormonal contraceptives on the immune system. And I'll break this down a little bit more in the next slides. So we set up to establish a pilot cohort with the help of the clinical core here at LGI. And we were interested in looking at three populations. Obviously, a control group, people taking hormonal birth control that has both estrogen and progestin, so a combined group, and then also progestin only. Unfortunately, we were not able to recruit any participants for this last group, so we had to stick with the first two, which is still interesting thing to look at. And thanks to the generous donations of multiple blood donors, we were able to isolate the circulating immune cells. And from there, we were interested in looking at two different things. And what really neat thing about the technology we're using is that we can do these at a single cell resolution. So for each individual cell that we extract from a sample, we can simultaneously study chromatin accessibility, which just shows us which parts of the DNA are open in each cell, and then gene expression, which essentially shows us what genes are being activated or launched. And another way to think about this is if your DNA were a cookbook, chromatin accessibility would allow you to see which recipes have been looked at, and then gene expression, you're seeing what has been cooked in a way. And we can integrate all of these data together, and we end up looking at something like this. So you can imagine this is data that has a lot of dimensions because there's a lot of variables, there's a lot of genes. We can condense it, and what we end up seeing here is each little dot is a cell, and how close two dots are or two cells are represents how similar or dissimilar these cells are. And because we know the gene expression of these cells, we can assign a cell type to each of these cells. So here broadly, we could identify here our T cells, our B cells, and our monocytes, or we can go into even more detail and break them up into really distinct categories that we can study. And I should mention these are all very preliminary results, and it's a small pilot cohort. But one of the first things we wanted to look at is whether birth control is changing the composition of immune cell types. So roughly looking bird's eye view of the immune system, what is being changed? And one of the interesting things that I wanted to highlight here is, for example, for natural killer cells, we do see a pretty significant decrease in the number of circulating natural killer cells in the group of people taking hormonal contraceptives. And this is something that was consistent across all our hormonal contraceptive samples. We are still investigating and working on the downstream analysis of these to be able to look at specifically which genes are being turned on and off differentially. And one of our crucial limitations for now is the cohort size, but this data is really important for us in that it's a proof of concept and allows us to generate more hypotheses that we can then use to apply for follow-up funding for what is a new research direction in the lab. So obviously in the future, we would want to expand the cohort and leverage this with more existing data that is generated in other institutes and by other labs. And finally, and I think this is something that is very exciting for me, we would want to include looking into hormonal replacement therapy because during menopause, hormonal replacement therapy is similar to hormonal birth control in the sense that you're using exogenous hormones to change these fluctuations, or in the case of menopause, when you no longer produce those hormones in your body. And in general, I think this opportunity has given me the chance to really see why research like this matters. It is very important, I think, to invest in getting a proper understanding in how these things might be affecting our health, what other things they might help us with, to have really better prescription guidelines. And funding like this that is flexible is critical to allow us to get these proof of concept data. And also personally, as a young scientist starting my PhD, I am extremely grateful to the opportunity. I have learned a lot during this project and I continue to do so. So thank you to the Spark program and everyone in the LGI advancement team for this opportunity. I really appreciate it. And also, of course, all of this work would not have been possible with the support of my mentor, Dr. Farhat Ai, or key members of the lab who have been very supportive and I've learned a lot from as well. The LGI Clinical Core, Gina in particular, and the LGI NGS Core, Susie, they have done so much work for these, and I'm really grateful for their work. LGI is a great place to work because of all the people that work here. So yeah, thank you to all of them and everyone who donated for the project as well, and of course all of you for listening.
Thank you, Laura, for sharing with us your fascinating project linking oral contraceptives with immune cell composition and characteristics. And I'd just like to stress to you all that Laura is learning what all well-trained scientists know, and that is that fluid timelines are a part of the very best science. And so now we can transition to our question and answer, our Q&A portion. So we'll return our wonderful speakers back to answer some of your questions. Amparo, and there's Laura. So the first question I have is actually for both of you, or for each of you. Both of your projects tackle such big gaps in women's health research existing today. What drew you to these questions, and why did you feel that they were worth pursuing?
I can start if you allow me, Laura. Yeah, for me, I think the first thing that got me into this field was how unstudied everything was. Normally, when you are a scientist and you think you have a great idea, then the next thing you do is go and search if somebody else have thought about this and try to pursue it. And normally, your idea has been already tried and failed. But in this case, when I was researching about this, I was really shocked of how little we know about a lot of aspects of women's health and sex-based differences. And talking about the impact, I want to stress how any advance in sex-based difference will be a step closer to personalized medicine. Anything that we learn can be applied to enhance medicine to both sexes, right? But also even anything that is only targeted to women's health is already targeting half of the global population, and also the half that bear the most impact on reproductive health. So hopefully the whole humanity will take advantage of any discoveries in those fields. Laura.
Yeah, I agree with Amparo, and I think also just looking at it from a pure perspective of just being curious about, but what is happening here, what is going on, and how can we understand better? I think that's what motivated me to try and start my project. And yeah, in the case of birth control, historically it is women who have borne the brunt of having to, take these medications for contraception, right? Or in the same way that a lot of people are saying this one-size-fits-all approach, some people that I've talked to have mentioned that for some gynecological conditions, that has sort of become the standard, and they sort of put a pause on that. It's like, oh, you have, PCOS here, take birth control, and it'll help manage symptoms. But, we're not really searching for these precise understanding that could help us find better cures and understand the causes at a deeper level.
Thank you both. I'm going to answer this next question coming from Amrita, who asks, are sex-based differences present at birth? And the answer to that question is yes. Before birth and at birth, our XX and XY chromosomes are immediately impactful in the development and the growth of our immune system. So there's really these impacts are felt immediately. They change over time, for example, through puberty and then through additional aging, but they are immediately impactful, to answer your question, Amrita. The next question is again to both of you, Amparo and Laura. How do you envision the discoveries that you're making now, today, shaping future therapies or diagnostics for women's health and beyond?
Laura, do you want to?
Yeah, if I can go first. Thanks, Amparo. Yeah, I think in general, the first change would be maybe helping out with prescription guidelines for people who are currently suffering an autoimmune disease or are worried that they could develop one. And in general, just having more information out there for the general public in understanding what is going on in our bodies. Like most medications, if you're treating heart disease and your medication has some side effects, it's usually worth it. But when it comes to contraception, whether a small risk of this or that is worth it, it's really going to be a personal choice because you're not fixing anything when being taken by healthy individuals. So that's where I think it would be really interesting to have more information.
So in my case, I would like to stay prudent and realistic, and we don't know how our lines of work will affect nowadays patient, but hopefully it will. And yeah, it will be amazing to have contributed a little bit to the lives of normal people. In my case, in the case of the use of T cells and understanding the role of T cells in reproductive disorders, the first thing that came to my mind is diagnosis. There are so many of these disorders that are really difficult diagnosis. Endometriosis, chronic endometritis. A lot of people are suffering a lot emotionally and economically because they don't even understand which type of disease they have. So the first thing, of course, always diagnosis. The second and hopefully next thing, therapies for that.
Thank you both. The next question comes from Bennett, and I'll answer. Bennett asks, are there other organizations exploring this space as well? And I'd really like to stress how proud I am and LJI is to be an early scientific leader in this research space in particular. But like any great idea, great science requires collaboration, it requires outreach, it requires working with others who are equally as talented and enjoying their talents. And for example, we work closely with WHAM, founded and led by LJI board member Carolee Lee. And WHAM's mission is to amplify investment specifically into the area of women's health research from both the federal and the private sectors. So you can see that, again, we're so proud to be one of the early kind of basic research leaders in this space, but we're committed to working with effective partners as well. So my next question is for Laura. Doing this work on women's health and sex differences, did it shift or change how you think about medicine and about research more broadly?
Yes, I think, first of all, how I think about medicine personally, it's really opened my eyes to how many variables there can be that can impact a person's response to a given drug, and how important it is to, maybe stop looking at things at the global level and be more personalized in our approach to medicine. And secondly, in terms of research, it's given me a really strong appreciation for all of the work that goes on behind the scenes for a lot of these things. It was my first time being able to be involved in a project from conception of the idea until, data analysis and all these things. So that's been, I think, a really important experience for my career.
Thank you. Amparo, how did philanthropic support for your independent project change the way that you approach your science as well as your career planning?
Yeah, having this program started from scratch, it's been absolutely amazing and rewarding. Also, you realize a lot of other things that normally your boss do, and now you have to do it. And I think that for both Laura and me has helped us a lot growing up as scientists. And speaking of my career, I am now fascinated with the world of women's health. And I will totally, if it's on my hands, to continue pursuing this field in the future.
Love that. I'm going to answer the next question from Robert, who asks, what is the role of menopause in affecting immune responsiveness? And this was addressed partially by Laura during her presentation. And I just want to stress that menopause is particularly complex because it is integrating at least two kind of major changes, that variables that would affect the immune system. And one is what Laura talked about, which is changing hormonal levels, which are decreasing naturally in terms of estrogen and progesterone, and combined with age, which also has an impact on our immune system. Adding another layer of that, as Laura mentioned, is that there may be the variable of hormone replacement therapies. So menopause is a time where the immune system is changing dynamically. And the response of a woman going through menopause is her response to bacterial and viral pathogens, and perhaps even her susceptibility to certain inflammatory conditions is dynamically changing. And sadly, we don't actually have very good metrics to fully understand how this is affecting the immune system, but that's what we're here for today. And I think that the challenge is going to be undertaken. So my next question is going to go to Amparo. And I wanted to ask this question, which comes from Barbara, asking, do you see male-specific T cells in menstrual effluent blood?
Yeah, it's a very interesting question. I don't know what you had in mind, either these T cells coming from microchimerism of potentially having had a male baby, or rather you were thinking of T cells that get into the female body through intercourse. In any case, with the analysis that we are currently doing, we cannot answer that. We are not doing chromosomic analysis, but it will be interesting to see. I will guess not a lot, and even in the case of the microchimerism for potentially having had a baby, normally is more studied and is expected to occur more in the other way around. So the baby gets T cells from the mother. But yeah, really cool. And yeah, hopefully we will find opportunity to study it.
And Amparo, kind of the second sub question, are you able to measure proteins in your material as well?
Yeah, we are also, apart from the cells, we are collecting the plasma, not serum, because we are not coagulating the blood. But yeah, we are also interested in understanding, well, our first goal is T cells, but hopefully we will also measure antibodies and any other proteins that in case there is some ideas or theories that we want to test. We are keeping also plasma from all of our donors.
Thank you. And I have one last question for Laura. Was there a time early on when you doubted that your project would work? And how did you move past this period of uncertainty?
Yes, multiple times, I think. I think I've just been really lucky in being surrounded with people who are experts in what they do and have been really supportive in me navigating a new field. And I have been really lucky in that sense. So I think I was able to move past that thanks to everyone's support, and having great mentorship and companionship from my lab mates. That has been the most important piece, I think.
I love to hear that. And, you know, we've all been there, and LJI is such a great place to work, and because of the people who are here. And I just want to echo that.
Definitely.
So thank you both, Amparo and Laura, for sharing your insightful work today, and really for reminding us how much untapped potential there is when we center on women's biology and sex-based differences in the immune system. And as you've all just heard, here at LJI, we're really keen on opening new frontiers in understanding the X and Y of disease, how women and men's immune systems work from the level of genes to individual cells, to the whole organism, to the person, to the individual level. And this focused research is just beginning to reveal why certain immune cells behave differently in women, contributing to higher rates of inflammatory and autoimmune diseases, or why men are more likely to face severe outcomes from infections. And these discoveries that we're making are already changing how medicine approaches diagnosis and treatment of a wide range of immune-related diseases. And of course, the next step is translating this knowledge that we're generating into real-world therapies that can reflect the real biology rather than assumptions or blind spots. So with sustained investment, the Center for Sex-Based Differences in the Immune System can build on work like Amparo's use of menstrual blood to uncover new clues about endometriosis, on Laura's exploration of how hormonal contraceptives are shaping immune cell behaviors. And expanding on these kinds of genomic and cellular studies, testing how treatments work across diverse populations, and designing interventions that interrupt patterns of disease before they take hold will move us closer to a future where medicine is precise, personalized, individualized, and effective, where care is guided not by averages, but what's really best for each individual and their loved ones at that current time in their life. If this vision resonates at all with you, I invite you to be a part of our community donors who are really moving this work forward in such an impactful, important way. Join us at lji.org/supportxy to fuel the next stage of women's health research, expanding studies like Amparo's and Laura's that you saw today, and turning discoveries focused on how diseases affect men and women differently into a new type of care, precise, equitable, and tailored to the individual. In addition to supporting the center, I just want to share a timely opportunity for those considering a longer-term impact. Thanks to the generosity of a current board member and their family, every new gift made to La Jolla Institute's endowment will be matched dollar for dollar until the remaining $700,000 in matching funds is fulfilled. Endowment gifts are invested to generate reliable, permanent support for LJI's mission year after year, fueling the type of bold science that you saw today, advancing our leadership, and protecting discovery from the ups and downs of traditional funding cycles. This match is a powerful way to double the long-term impact of your philanthropy while securing the future of research that changes lives here at LJI. Learn more at donate.lji.org/endowment. Just want to stress that it's your curiosity, your questions, your investment of your time and your support that makes all of this work possible. And together we can change the future of health for everyone. So thank you so much for joining us today for Live from the Lab, and we look forward to welcoming you back soon.